To understand the Cholangiocarcinoma Screening and Care Program, Cascap, and why it's so important, we first need to understand what liver fluke is. Liver fluke is a parasitic worm that has a very complex life cycle, with several stages of development across multiple different animals. The adult form of the parasite exists in the bile ducts of humans, and to a lesser extent, dogs and cats. The story of why Cascap exists begins back in history, over 100 years ago, when liver fluke infection was discovered during the post-mortem examination of 2 prisoners at a jail in Chiang Mai. It was later identified and reported as a species of liver fluke named Orpisticus viverini, or OV for short. In 1965, a scientist identified that freshwater fish from the carp and minnow family, collectively called cyprinid fish, are the intermediate host of OV. In 1970, the transmission of liver fluke to humans via the consumption of raw or undercooked cyprinid fish was discovered. In the north and northeast regions of Thailand, it is part of tradition to eat cyprinid fish that is raw, like koi pla, or slightly fermented, such as pla ra or pla som. The dishes prepared with these fish are still very popular and are regularly consumed by many people. These fish preparations are the primary source of OV infection in the people of Thailand, as well as other Mekong countries. When the fish containing the encysted larvae of OV are consumed, the larvae exist in the beginning of the small intestine called the duodenum and migrate into the bile ducts, where they mature to adult form and each begin laying as many as 500 eggs per day. These parasites can go undetected and remain living in the bile ducts for as long as 20 to 30 years, not presenting any noticeable symptoms. OV is classified as a Class 1 carcinogen by the International Agency for Research on Cancer. OV infection, or opisthorchiasis, causes inflammation as part of the body's immunological response to the infection. This inflammatory response is the principal mechanism for fluke-associated cholangiocarcinogenesis. The inflammatory response triggers chemical reactions which produce free radicals and leads to chain reactions that damage the cells in the walls of the bile ducts called cholangiocytes. This oxidative stress caused by infection leads to DNA damage, tissue remodeling, and the alteration of gene expressions. Although it takes 20, 30, or even 40 years for a person at risk to develop CCA, no stronger link between a human malignancy and parasitic infection occurs than that between cholangiocarcinoma and infection by OV. The northeast region of Thailand has the highest incidence of CCA worldwide, with OV infection being the single biggest risk factor for developing the disease. More than 50% of the people diagnosed with CCA are suffering from the late stage of the disease, with as many as 20,000 people that die each year. Although it is a very aggressive cancer, it is also a silent disease in that symptoms often do not present until the cancer is already in an advanced, usually untreatable stage. For this reason, early detection is crucial for the long-term survival of people with this disease. Cascap operates as a mobile screening program, touring across the north and northeast regions of Thailand to provinces with high-risk group populations and provides free CCA screening. The people that Cascap benefits most are from rural, impoverished areas with limited access to medical care and are usually unable to afford the cost of medical attention. The program brings skilled healthcare professionals and state-of-the-art imaging technology to the people who are most at risk for this disease. Cascap is also an advocate for CCA awareness and provides information about the disease to everyone. Cascap is one of the programs launched for the occasion of the 50th anniversary of Khon Kaen University, commemorating 50 years of social devotion. The program was fully implemented in February 2014 with the aim of providing long-term screening with follow-up to the CCA risk group population, to develop a high-quality database of compiled information about CCA in the region, and to develop a sustainable cooperative network of hospitals and healthcare providers. The database is called ESON Cohort and is fully operational, with more than 23 million people consented and registered. Over 500,000 ultrasound examinations have been performed and recorded. There are 22 general hospitals, over 350 community hospitals, more than 3,000 district hospitals, and over 6,000 registered care providers. ESON Cohort addresses the need for a national database to monitor and evaluate programs for the prevention and treatment of CCA. It is a web-based application that facilitates scaling up of tools for those purposes. The datasets form two main cohorts. First is the screening cohort, comprised of the risk group population who underwent at least one occasion of liver fluke screening or CCA screening using ultrasound examination. The second is the patient cohort, or the Cholangiocarcinoma Cancer Registry, comprised of patients diagnosed with CCA. Data quality assurance is performed by a team from the Data Management and Statistical Analysis Center, DAMASAC. This independent monitoring and auditing ensures that the data is research quality. There are 11 projects of Cascap, which together form the master project called Fluke-Free Thailand. Fluke-Free Thailand aims to resolve the problems of liver fluke and CCA from the primary, secondary, and tertiary levels. In 2016, the National Research Council of Thailand organized a program called Thailand Research Challenge and subsequently awarded Cascap with a grant to fund the Fluke-Free Thailand project. The first project is aimed at food safety and focuses on education geared towards the prevention of liver fluke infection. The project teaches people how to avoid infection by preparing foods in ways that eliminate the risks and encourages the innovation of traditional dishes using safe ingredients to develop fluke-free foods. The second project is aimed at innovating the techniques for diagnosing OV infection using rapid testing methods. Traditional methods of testing require the collection of feces, which is then examined by experts to identify the presence of eggs, and is an altogether difficult process. The project has led to the development of a rapid testing kit using urine and the test strip. It is a simple-to-use test which provides accurate results within 15 minutes and is now in the stage of being mass-produced. The third project addresses the need for treatment in cats and dogs, which are also a host for OV. This problem requires immediate attention due to the very high number of stray cats and dogs in Thailand. The project also works to educate people and encourages pet owners to have their animals treated. The fourth project aims at improving the sanitation systems to prevent fluke eggs from being released into the environment. The focus of this project is to work with Thailand's Ministry of Interior to develop the infrastructure of sanitation systems in areas where they are absent or inadequate. It is equally important to encourage changes in sanitation practices to ensure that raw sewage is disposed of in a safe manner. The fifth project addresses the need for a centralized database, networking healthcare providers and health institutions, and serving as a shared database for patient information. ESON Cohort was developed for this purpose and is designed to support the management of care for patients and to help capture the data needed for the continued research of OV and CCA. The sixth project focuses on education for the awareness and prevention of liver fluke and CCA. The project is driven to provide people with the understanding to make better decisions about their health and to improve the raw attitude towards behavior that puts them at risk. The seventh project addresses the need of routine screening for early detection of CCA in people at high risk for the disease. The aim is to help healthcare providers adopt screening protocols in areas where there are high-risk group populations, and to provide training to improve the standard of care for diagnosing and managing CCA. Surgery is currently the only curative treatment for CCA. However, it is often diagnosed in an already advanced stage not accessible to surgery. The 8th project is focused on clinical research to improve the efficacy of current treatment methods, to find additional treatments, and to develop ways to improve survival rate. Many of the ways to combat OV and CCA require new policies or changes to existing policies, which requires the support from many sectors of government. The 9th project is geared towards policy research by developing a specialized team that understands policy making to find the most effective ways that government can adopt and implement these policies. The 10th project focuses on the study and analysis of OV and CCA in terms of humanities and social sciences. It is necessary to study and understand the anthropological aspects of OV and CCA to develop a sustainable solution for disease prevention. The 11th project is aimed at developing a Geographic Information System, or GIS, designed to serve multiple functions. GIS will aid patients as a resource to connect with clinicians who can assist with palliative and end-of-life care. The system also benefits research by tracking regional occurrence rates of OV and CCA, and can also be used to track awareness programs to help reach areas most in need. Fluke-associated cholangiocarcinoma is a preventable disease, and although it is an ambitious task, Cascap takes ownership and is devoted to solving these problems. The issues of liver fluke and CCA have continued for far too long, but with the support of government, private sector, and the international community, we will succeed at overcoming these problems and save lives.